That was wonderful, Dr. Kumar. We definitely, I think, could have heard about another hour on the pharmaceutical side if it was time. I know that's a subject that could fill many hours. So thank you for making it available to us in the PowerPoint presentation. We really all have to give our hearts to a person who comes at the end of a long day like this and has been so patient, and everybody just really needs to give them your hearts and minds. I certainly have a world of respect for them. I've been in that position before, and that's a very difficult position to be in. Dr. Qian Wang received his PhD in chemistry from Tsinghua University in 1997, and after his postdoctoral studies at the University of Lausanne and the Scripps Research Institute as well, he began teaching as an assistant professor for the University of South Carolina. He then published some 70 papers and has been awarded National Science Foundation Career Awards. He's also been named the Alfred P. Sloan Research Scholar. He's going to speak to us today on self-assembly of nanobiomaterials for tissue engineering. Dr. Wang, thank you very much.
So first I want to thank Dr. Rothblatt, invite me to give this nice talk. And it's my honor to join the praise speakers in this session this morning to give some study in our group. But I have to say one thing before I continue on, is it's really not difficult to be the last one. Since I have hundreds of slides, so I can take whatever I want, and then give enough of the nanobiomaterials as appetizer and the dessert. Okay, I'm just teasing here. So today I want to tell you something is the work in our lab. And I want to join Carla just mentioned the computation. I want to bring some new particle for you if you haven't heard about this plant virus. So in my group, we're trying to work on the plant virus, and we call it nanoparticle. And such kind of particle is either the rod shape or maybe spherical shape. We can purify from the plants in gram or kilograms quantity in very cheap and inexpensive way. Very easy. And such kind of product, this kind of particle, we can selectively modify them, give a very precise precision. So what I see, which I show here, is the structure. So the particle we are talking about here, each of them is a couple million or tens of million Dalton. However, since we know the structure to near atomic level, so it's very easy for us to modify such kind of particle to a near atomic resolution. So that's something, if you want to give the computation, I don't want to say the others, but the precision or the functionality in terms of the radius selectivity, I think our particle definitely will win the computation. And we know the structure to very fine resolution. But I think the focus on our study really is on how to assemble such kind of particle to give the further structure, either the one-dimensional array or two-dimensional array or three-dimensional hydrogel work. And finally, I will go back to my real interest, is how to use such kind of materials in order to map how the cell will communicate to a certain programmable scaffold. Okay, so the virus is something very interesting. In case you don't know anything about the virus, just let me give a very brief introduction about what is a plant virus. So first thing you want to learn is plant virus is very safe. Just like this morning, this lunchtime, Dr. Blumberg talked about, so the virus have a very specific host. For the plants, no matter in Canada or China or US, you go to the grocery, you buy something, you eat it, you have the plant virus in it. However, it's very safe to the human being. So that's something if you use it as biomaterials, at least for the students, they are happy. They don't need to worry about the infection and the other thing. And in general, for the plant virus, there are two parts. So one is what I show you here, the two typical spherical particle. One is the copy mosaic virus, another is the turnip yellow mosaic virus. So you see the size of these two, they all are around 30 nanometers. Just as Dr. Rothblatt said, it's a nano device, nano machine produced by the nature. And there are two parts. So one part is the outside I show here schematically is composed of the protein structure. So for one particle, there are about 60 copies of such kind of, we call the asymmetric units, assembled together, give the final icosahedral structure. And inside, there are two types of genome. And for the one of the genome, this is the genome of the cPNV. So for the genome is the RNA genome. And for one part, they express or represent whatever information on the capsid or code protein here. So that's something very unique. If you have a synthetic particle, it's really difficult to control the functionality at a given position. However, as a bioparticle, since we know the genome here, so we can do some kind of simple molecular biology manipulation. We can insert or delete some sequence here in the bacteria, E. coli. And then what happens is we can easily generate another type of mutant virus or some kind of improved virus with certain modification at a given position. And that also can translate into the reactivity. I will show you one slide later. The same thing as the rose-like particle I show here is the tobacco mosaic virus. It's one of the earliest viruses has been ever discovered. And for such particle, the length is 300 nanometer, and then the diameter is 80 nanometers. So for every given particle, if it's TMV in the same plants, they have the same size and the same shape. So that's another thing I think we can win the computation is for when we talk about the monodispersity, what we have here is really monodispersed. They're all identical to each other. And that's very crucial for one of the applications we are doing is how to use such kind of identical size and then well-defined structure. We can control the assembly pattern. So that's something I can show you. Some work we can do it very easy with our materials. So what I'm going to do is I want to give you a very brief the protocol of how to purify it. So how these plants grow in my lab, for example. So if I have this space here, about 2 square feet or 3 square feet, I can easily grow about 2 to 3 kilograms of the wet tobacco plants. And from 1 kilogram of the tobacco plants, I can get about 2 grams of the particle. So the overall cost, even I pay my students fairly high salary, but still is less than $200 per gram. And the purity is 97% pure. So the difficulty of the purification is none. So we can get these leaves, put in the freezer, and then go to the sales. Of course, you need to buy a blender and buy some cheesecloth, add some water and ethanol inside, blend it, and then buy a simple filtration, add some sodium chloride inside. You can get a pure particle. Very simple. So the longest time is really how to grow this particle in the plant room. Of course, some of the students are really good. Lisa Alexander, very happy. What I want to put here is, you see, you don't need to have the gloves working with the plant virus. It's very safe, plan on that. So it's a green synthesis in the laboratory. You can get cheap starting materials. So that's one of the major reasons. It's very easy to get large quantity, can easily manipulate the surface property. So that's the reason that there are many groups growing, working on this lab, working on this kind of area. I think, before, about 5 years ago, I started in South Carolina. So I put these slides together. There are only 5 big groups. I put some like Johnson and Finn and Mary Manchester. They are in one group. So there are only 5 groups, including my group, working on the virus related to chemistry and the materials. But now, I organized another meeting for the ACS meeting, in this Philadelphia. I count about 50 groups very active in the field already. So it's a growing area. Now, in my lab, we try to use this ocean of big particles from the natural or nanomaterials from natural, and can do many applications, including the drug delivery, the conductive nanowire, catalysis, and the biocomposite materials, many things. But for today, I only want to introduce one set of story, just give you a concept, how we can selectively build up some kind of functionalized particle, how we can control the 2D, 1D assembly, especially 1D assembly. And finally, I want to tell you how we can use such kind of structure to see the cell culture on that one. But before I start to tell the story, I want to show something very crucial, is why I'm interested in this work. That's really come from one of the early papers in 1995. So it's one Nobel Prize laureate. The name is Rolf Zinganaga in Switzerland. And he published one paper. The name is, in science paper is Immunology Learning from Virology. So in this beautiful paper, he talked about, without any solid experimental data, he gave this hypothesis is the immune cell and the virus and the other pathogen, they have this co-evolution, okay? And the interaction really caused how the human built up the immune system, and also how the pathogen can adapt to infect human. That's a really elegant conclusion in that kind of paper. And what I learned from that paper is that's very interesting because when you see the cell interaction or cell extracellular matrix interaction, there are so many level interaction getting involved. So one level is, of course, we talk about in the small molecule. Many of the speakers talk about we have the ligand, have biomarker, and can recognize the cancer cell. Is that true or not? Maybe not. Yes, the most common is the targeting unit, the folic acid. They can only target a certain type of the special cancer, and also they have a lot of false targeting. The reason is like cancer, there are so many biomarkers, but you cannot really tell one biomarker is universal useful. So how to identify this biomarker fundamentally will go back to one question I'm asking here. So what kind of interaction or how it can impact on the interaction between the cell and the cell, or cell and the cell extracellular matrix? So when we go down the chemistry, I'm a synthetic chemist. I want to just put everything in the structure. What I show here is first, is a small molecule, how the mono, with a receptor like RGD with the integrin, how 1-1 interaction. And then this kind of interaction can really, you need some kind of polyvalency. This polyvalency is very crucial. Philip talked about the carbohydrate. Yes, polyvalency is one thing of the carbohydrate, how to interact with the lecithin or some kind of receptor. But that's not all. When you have such kind of interaction, they want to have this kind of self-organization of the membrane, cell membrane, and then to adapt the conformation, like here, if it's a virus or pathogen, they really want to reorganize such kind of structure. That's the elegant paper by the Laura Kisling in Nature a couple years ago, talking about how they can adapt the information from the environment and reorganize the cellular receptor. And finally, we're not talking about one of them. We're talking about multiple receptors here. And eventually, we're talking about not only one cluster, it's a multi-cluster. They have to have certain type of distance. That's something very famous virus is the adenovirus 2 and also the foot-mouth disease virus. So when they target the human cell, they not only need one cluster, they need many clusters, many receptors come together, have defined geometry in order to give the cell recognition there. So now you see how many we talk about. We talk about one very simple interaction. We have this cluster and cluster distance here. And finally, we have to add some other thing, the mechanic and the physical properties inside. So in other words, if you really want to learn how to learn a cell-cell interaction, or how to find out the cell can recognize, interact with the cellular matrix, you have to know the chemistry, the geometry, the spatial distribution, and also the physical properties. So that's just impossible to use a normal or natural ECM, extracellular matrix, to do it. So we have to design something can have this kind of precise control from the sub-nanometer that I'm talking about is a single interaction to a big micron size. This is the size of the cell. And also you have to program the mechanical property and the electronic property inside. And that is the reason why I think virus and also similar bio-nanoparticle can give very nice scaffold for that kind of work. So I'll go through my story about I show you how to functionalize so we can control the sub-nanometer precision based on the simple bioconjugation chemistry or maybe some kind of genetic modification here. And then I will go through how the assembly, we can go to the big scale. We can go from the nanometer to micron or millimeter. And finally, I will tell you one story about cell response. So modification, very easy. The protein chemistry here, many things for organic chemistry. That's piece of cake. We can do a lot of work. So in this special work, Hannah Banghui in my lab collaborated with Xuefei Huang and MD Fin in Scripps Research Institute. Sorry, I'm a little faster now. So how the copy mosaic virus. So what we have done is we use a genetic method to modify the surface. So in this case, this work is by the Fin and Johnson. So they have generated this kind of mutants. And then you see here, the distance of the two ligands here is defined based on the genetic information. For the red one here is a 28 angstrom, the two ligand. And for the purple one here is 50 angstrom. Okay, so now we are talking about how the big particle, 30 nanometers, however, we can control the ligand at 2 or 5 nanometers, very precise. We can go to even more defined than that one or more broad than that one based on the genetic information we have here. So what's the big deal here? So the big deal is you see here, we put the MG Fin and the Johnson, they'll put a nano gold on the surface, and then they can define this kind of distribution here. That's something I want to put in the computation is, so of course there are some other things the bioparticle cannot compete, like physical properties, magnetic properties. But for the precision or the functionality here, so no other particle can have such kind of wire control here. And this kind of control can be translated into many applications. Like one work we are doing with Xuefei Huang is we put some kind of special, very simple antigen, it's a carbohydrate-based antigen on the CPMV. However, the distribution here is almost the same thing, but it's another mutant. We have the double mutants there. So we have 120 of such kind of mutants, such kind of antigen on the viral particle. And we find out, compare with the immune response from the rat. So if we use a single thing here, single antigen, Tn antigen with normal protein, compare with our virus conjugate, so this one is about a 10,000-fold more response come back. So it's much strong. And let's go back to the work by the Tsingenaga. Yes? So this kind of special pathogen or this kind of array alignment, this defined position here, will really cause the immune system have better, stronger response. That's one of the applications I can show you. The precise define the ligand distribution here have a lot of applications. So that's another big file here. So basically this is already very, very well organized and very well established the system there. So by MG Fin, Johnson, our group, Matt Francis, many groups have contributed to this area. So now we can put all kinds of functionality. So in my group, we put all the stuff here on the viral particle at the given position we have. So that's something I want to tell. The first information is we do have such kind of scaffold. We have the chemistry. We can have all kinds of functionality and at a defined position. That kind of precise to something like a sub-nanometer level. Okay, that's the first thing. The second thing is that we have this kind of particle since they are identical. So many chemistry or many physics developed for the colloidal chemistry, we can borrow for our particle here. In particularly, we can use many simulation to guide our assembly work because they are identical here. Like I show here, we can easily assemble the rod-like particle to a one-dimensional nanowire or two-dimensional array, and the spherical particle can give the two or three-dimensional structures. Due to time issue, I just give you two examples about assembly. So one is how to build up the two-dimensional array here. Okay, so we use a very simple strategy. So we use water and oil, and then you have this kind of particle at the inside. Let me show this slide. So you have the water here, chloroform, very common organic solvent, and then you just have this one, just shake it. Okay, and what happens is you form the droplet. If we don't have any bioparticle inside, so what you see is in 2 minutes, you have the layer separation here. Okay, you have the two layers, water and oil. But if we have the bioparticle inside here, this green fluorescent dye modified bioparticle inside, so you have this small droplet here. I have the confocal microscope here. So for each droplet, they are very stable for months, for years. The reason is because the bioparticle sit along this edge here. You can see here the one edge of the macroparticle. They act as something like a macro surfactant to stabilize the interfacial energy. Okay, so they form this droplet. And if you don't shake it, you just put on the top like that for 2 months. So what will happen? No, initially we don't shake it. So what happens is, so we can easily form this kind of very well organized two-dimensional array, either with the spherical particle here or with the rod-like particle. Simple process. We shake it or not. Use this interfacial energy. They self-assemble, give us a structure here. So what's the big deal there? Okay. So there are many applications I will show later, but one thing I want to show is so we can replace such kind of organic solvent with polymer. So very simple one, P4AP, polyphenylpyridine. Everyone know that one. Even I'm not a polymer scientist, I know this one. So buy this one from the company, and we shake it with water, and the Tali has done this work. So what you have done is this kind of beautiful raspberry structure. And that solves one big problem is there are many groups working on using the viral particle, plantavirus or bacteriophage to do drug delivery. But they always use the single particle here. The particle size itself defines the space inside. So you don't have many space to put drug inside. So in our situation, what you have is we can control the size based on the ratio of the particle and also the polymer. So we can control from the 100 nanometers to 500 nanometers. And for the 100 or 200 nanometers one is ideal for the drug delivery. And in particularly, we choose the special P4VP derivatives. It's very sensitive to the pH. So if I have this one at pH 6 to 9, they are very stable. But whenever you hold about 5.5 to 5.8, it dissociates. And we can how we haven't done the detailed quantitation about how much doxorubicin or the other drug get inside, but we can see clearly uptake is much more than a single particle. So what we have here is we combine the two advantages. One is the advantage of the special bioparticle, because we show you, and I show you, we can control the ligand display. All kinds of ligand we can do it by the genetic way or chemical way, and then you can do a very efficient targeting. But on the other hand, we use this kind of polymer strategy. We can put the drug inside, and of course, it's very pretty also, try to push this paper out. That's something that we can combine the structure feature and the chemistry of the bioparticle together, learning from the other folks, from the other particle, and we can design something useful for the drug delivery. But that's not really my interest. My interest is really another thing. That's another beautiful picture I'll show you. So another thing we can assemble is, we can do the 1D assembly. So this is a rod-like tobacco mosaic virus. So remember the length is 300 nanometers. So in order to mimic the extracellular matrix, so if you know the structure, the ECM, so the major component is the fibronectin or collagen fiber. So you do need to form the fiber structure. And what we want is we want to do something artificially. So that's why we start to use the rod-like particle TMV. And what we design is maybe you can control them to give the head-to-tail assembly. So that's already been done a long time ago by Clark. They know, if you control the pH, you can observe something, like here is 600 nanometers. So the two particles align together. But it's really difficult. It's really fragile. And also when you control the concentration, you always give some kind of parallel alignment. So this one will win the game. So how to control it? So Zhongwei knew in our lab do something quite simple. So we add some kind of small monomer inside. There's some kind of recognition on the surface based on the charge-charge interaction. And then we do the in situ polymerization. So we just like you have two chopsticks come together and use the special scarf to wrap it, or scotch tape, whatever you want to call it. So Zhongwei, this is the original bioparticle of TEM, AFM, and then if we add some aniline, simple starting material, and then ammonium persulfate inside. So what happens is you align the particle together, give this kind of quite nice dispersed nanowire. And you can really easy to pick up one single wire. Of course, when you talk aniline, polyaniline is conductive, so we can easily get this kind of conductive polymer come out. We can do some nanosensing work. But in my interest, so I really want to do one thing is I want to control this kind of assembly, go to 1D, 2D, 3D, or the other structure. So we don't see this one here. So I call it the Chinese noodle, or maybe someone want to call it Italian spaghetti, whatever. So this is really like nano noodle. Sorry for that. Yeah. So we have this really similar to the extracellular matrix. So we have this fiber structure aligned together. So what's the next step? So that's something I want to show you. So this kind of thing, if we have the alignment, like here what I show is another bacteria M13, easy convertive alignment. We can give this kind of two-dimensional alignment in a big area. So we can have the cover slip, all areas like that. And then if you grow the cell on that one, with normal cover slip, you see the cell grow any directions. But now, with the fibroblast, they all grow along the direction of the alignment we have here. So this kind of nano macro feature can translate to dictate the cell growth. The same thing here. See, this is a CHO cell. You can see all the cells align along the direction of our viral scaffold. So what's the big deal there? So we have collaboration with Xujin Wang in MUSC. So he's interested in this central neuron system repair. So what he's struggling is how to fabricate something inside the special polymeric nanotube. Macron tube. So you want to use the macron tube to link these two spines together and to force the neuron grow inside. However, there are big problems to do that. The reason is you need an even smaller feature. You have to have something like 100 nanometers, this kind of feature, this head, all aligned along one direction to force the work. The problem is, there are some work like, Charles Lieber or some other group have done a wonderful job in the lithography to do work. But now what we are talking about is not a chip or some beautiful TM. We're talking about really a fiber this long, this kind of fiber this long. All the inside, you want to align them to the feature. It's not trivial work. That's something we collaborate. We try to do the same thing. We translate this kind of alignment, this on surface and in the tube. Now we are trying to see whether we can put this one in the pig to see what's going on there. So it's still ongoing project. To finish my story, I want to give one example doing our work is we use the bone marrow stromal cell. So this is adult stem cell, and there are many possibilities. Can go to the osteoblast for the bone formation, can go to the cartilage, can do muscle, whatever. So in our lab, we just start to do one thing is we want to see how our scaffold, I show you this kind of Chinese nano noodles, how that kind of noodle can play with, because we have this polyvalent delivery system. We have this kind of functionality. So whether, go back to my first, the intention, how this kind of nano to macro scale assembly can impact on the cell-cell communication. In this particular situation, how it will impact on the adhesion, spreading, proliferation, differentiation, and mineralization. So to make the long story short, Gagan Deep in my lab in collaboration with J-Pulse can see, our scaffold is biosafe, and based on very simple RGD modification, we can have the cell grow on the surface, and then they form, this is the alizarin red staining. So the red color show we have calcium. So in other words, we already can translate to from stem cell to the final osteoblast. So very nice. And there are confocal microscope. Let me show this one. So that's the beautiful work by the Gagan, is use the real-time PCR to really see whether we have the gene upregulation. And there are many data in it. The only thing I want to point out in this figure, so you can see this is the most important gene, is osteocalcin. So this gene, normally you want to see such kind of distribution here, means the peak number is higher than 300-fold than the control. It means you have the very good differentiation. That's just bottom line. There are more detail on that one. And you want to show this kind of shape. So what we see here is that in day 14, is one of our TMV, tobacco mosaic virus-based scaffold, we can already finish the differentiation. If you use normal tissue culture, all the hydrogel, whatever, from the commercial available source is 21 days to 28 days. So this nanofacial can upregulate the differentiation to the cell. And also very important is we can control that. This is something I want to show here. So if you use a rod-like particle, we form this kind of big thing. This is a centimeter long. These pieces. All the cells not only form the bone, but also they migrate together, give this kind of layer by layer structure. However, when you use the small spherical particle here, 30 nanometer particle, so they form this kind of island-like structure. So this brings two big questions. One is why we can have the upregulation. Second is how this kind of small feature here, the 30 nanometer, and what I show here is about 2 millimeters. So how this small feature can impact on the big cell-cell communication, eventually can cause the big morphology change of the final cell, this whatever bone, whatever you want to call it. How does it happen? It is very crucial to control such kind of cell aggregation. But what's the bottom or basic principle behind it? That's still something I think we are trying to solve in the next couple years or maybe decades or whatever. So that's my group. And the funding agency, many collaborators, and I have, unfortunately, this is the students in my lab, very nice students. And thank you very much for your time.